Hi there. This is a quick start tutorial meant to walk you through the basic functions of CalR version 2 and to help you get a better understanding of how to use it. This video is also a good tool for those who have used CalR version 1.3 in the past. If you have any questions about other functions or are confused about a specific part, please refer to the tab-specific videos under the Help tab. The Input tab is the place where all data is uploaded. Click the Browser button under the number 1 and upload your CalR file. This file has data from the calorimeter. You can also drag and drop the file into the empty space. Click the Browse button and upload your session file. Go ahead and click on the Time Plots tab. To begin visualization, let's look for the metabolic variable drop-down menu. Select which metabolic variable you are interested in. The first variable we'll look at is oxygen consumption. The example we are using is an experiment that involves treating mice with thyroid hormones and seeing their effect. Click Generate Plot. The graph we see right now is data of oxygen consumption for 8 days. To smooth the data and make it easier to understand, change the moving average to 20, which shows 3-minute data and 1 hour of smoothing. From this graph, we can see that thyroid hormone increases oxygen consumption over time. Let's see the metabolic rates on the last day of the experiment. Use the sliders or type in the start and end hour to control which hours you are looking at. In our case, 163 to 187, and click the Generate Plot button again. Every time you make a change to the hours you're looking at or removing outliers, remember to click the Generate Plot button again. Download the session file to save the metadata so it is easier and faster to visualize the data next time loading it into CalR 2.0. Scroll down to see the option to plot individual animal hourly plots. Click Yes and then Generate Plot. To the right of the Generate Plot button, you have the option to download excluded data points. In other words, any values removed by Remove Outliers will be downloaded here in a CSV file. There is also an option to download the independent hourly Prism file, which contains data averaged and arranged by hour in a GraphPad Prism friendly format. Scroll back up and click the Regression Plots tab. Let's choose the metabolic variable to go on the y-axis, energy expenditure. We can choose whether we would like mass or physical activity, otherwise known as the covariate, on the x-axis. In our case, let's choose mass, and choose the time of day, total. Click Generate Plot. You'll see the R-squared values under the group names. Additionally, in the upper left corner, you will see the mass effect, the group effect, and the significance of the result in the stars. Click the Analysis tab. Then choose the covariate you're interested in the drop-down menu, total mass. Click Run Analysis. This is the statistical analysis of your experiment. CalR will examine the data for significant interaction effects between mass and mass-dependent metabolic variables, including food consumed, water consumed, energy expenditure, oxygen consumption, and carbon dioxide production. If an interaction is detected for any metabolic variable, then the GLM results are displayed. If the interaction effect is absent, the model is rerun and the ANCOVA results are displayed. For mass-independent variables such as locomotion, total distance in cage, RER, locomotor activity, body temperature, and energy balance, we perform the ANOVA test for different time periods. Overall, this experiment shows that treating thyroid hormones increases oxygen consumption over time and is statistically significant. Once you're done with your experiment, and if you would like to share it with your community, please refer to the CalR repository tab. If you are interested in other tab functions, please take a look at our other videos under the Help tab.